We learned last time about the cell division cycle and how a complex between cyclins and the respective cyclin-dependent kinases can regulate progression through those division cycles. Today I want to do a bit of a deep dive into what, in addition to cyclin binding, can regulate CDK kinase activity. Recall that there are many different CDKs, and those CDKs are active at various points within the cell division cycle. In each case, a CDK must be bound to a cyclin in order to be activated. We'll focus the talk today on mCDK with a little bit of diversion toward the end into sCDK, but realize that the mechanisms that we're going to talk about with respect to mCDK and sCDK can be more broadly applied to all of the cyclin-dependent kinases. Recall that the cyclin-dependent kinases are typically present throughout the cell division cycle, but they only become activated after the levels of their respective cyclins rise so that the complex can be formed. The concentrations of cyclins are going to vary throughout the cell division cycle, with sometimes levels being high and other times concentrations being low. I'll start by talking about what turns cyclin-dependent kinase, specifically mCDK, off. If we look at this graph that we saw before, we see that mCDK activity is high only during mitosis. We also see that mCDK's cyclin, M-cyclin, increases concentration slowly over the course of interphase, peaking during mitosis. The concentration of M-cyclin rapidly falls at the end of mitosis, thereby turning off CDK activity very rapidly. How does this occur? Starting at the left-hand side of this slide, we see that mCDK is active. It is bound to its M-cyclin. So that creates the MPF, or mCDK M-cyclin complex. We know that it's active because of the red glowy lines that we see on the cyclin. This is during mitosis. Moving a little bit to the right, we see that there is ubiquitinylation of cyclins. So the transfer of ubiquitin peptides to the cyclin. Recall from earlier in the semester that polyubiquitination is a signal for transfer of a protein from the cytoplasm to the proteasome where the protein is degraded. In this case, ubiquitination of cyclin occurs via a protein called APC, anaphase promoting complex. APC is actually activated by active mCDK. So the active mCDK is responsible for activating APC. APC in turn ubiquitinates M-cyclin. That results in degradation of M-cyclin and inactivation of mCDK. In other words, mCDK is responsible for decreasing its own activity. So this rapid decline in mCDK activity is a result of mCDK activity. While it's fairly easy to see how mCDK activity can decrease so rapidly as a result of the degradation of M-cyclin, it's perhaps a little more difficult to understand how a relatively slow rise in M cyclin concentration throughout interphase can produce such a rapid increase in MCDK activity. In other words, the concentration of M cyclin is clearly correlated with the decrease in MCDK activity, but it's not as clearly associated with the increase in MCDK activity that occurs at the beginning of mitosis. That is because other things are involved in regulating the optimal activity of MCDK. Looking at this diagram starting at the left, we see that MCDK is inactive, and it's inactive because it is not bound to M cyclin. It binds to M cyclin, and in the center of the screen, we see MCDK bound to M cyclin, but it's still inactive. The reason that it's still inactive is because it has served as a substrate for the inhibitory kinase, WEE. WEE is a protein kinase that transfers a phosphate group to MCDK, rendering it inactive irrespective of whether or not M cyclin is bound. Like all phosphorylation, phosphorylation by WEE is reversible, and it's reversible by the activity of a protein phosphatase. In this case, the phosphatase is CDC25. CDC25 can remove the inhibitory phosphate from MCDK, and if MCDK happens to be bound to cyclin, now we have active MCDK. So the rapid increase in MCDK activity results not from only binding to M cyclin. As the concentration of M cyclin is binding, is increasing, certainly that M cyclin is binding to MCDK. But MCDK is phosphorylated by WEE, and it is not active. It's only after that inhibitory phosphate is removed that MCDK becomes optimally active. Removal of that phosphate, that inhibitory phosphate, is partially responsible for the rapid rise in MCDK activity. One other thing that I'd like to highlight here is that CDC25, the phosphatase that removes the inhibitory phosphate put on by WEE, is also phosphorylated in order to be active. What phosphorylates CDC25? Well, you might have guessed that it's MCDK. Looking at this image on the left-hand side, we see inactive MCDK, and it's inactive even though it's bound to cyclin because it has been phosphorylated by WEE. In this image, it's showing 2 phosphates. I actually don't know how many phosphates or how many different amino acids can be phosphorylated by WEE. In any event, the phosphorylated MCDK, even though it's bound to cyclin, is inactive. It's only after those phosphates are removed on the right-hand side that we see active MCDK. MCDK can phosphorylate CDC25. When it does, CDC25 becomes active and therefore removes the phosphates from MCDK, the inhibitory phosphates from MCDK. What we're looking at is an example of positive feedback. Certainly, it's not the same MCDK that is phosphorylating CDC25 as the one that's being activated. What happens here is that some small amount of MCDK becomes activated, and that small amount of MCDK goes on to activate CDC25, and that active CDC25 then is responsible for activating even more MCDK. So we have this positive feedback loop where some active MCDK is responsible for producing a lot of active MCDK. And again, this is the rise that we see in MCDK activity. If that were all that were involved, it would be great. But in fact, we have another aspect of regulation. On the left-hand side, we see that active MCDK is bound to cyclin. It is not phosphorylated on the inhibitory sites by we, but rather is phosphorylated by another protein. We'll look at that in the next slide. That adds an activating phosphate. So we have active MCDK that is bound to cyclin, not phosphorylated by we, but phosphorylated by an activating kinase. In order to turn off MCDK activity, we have many different options. We can remove its activating phosphate, we can add an inhibitory phosphate, or we can degrade M-cyclin. Here we see a summary of MCDK activation. MCDK throughout the cell division cycle is present, but it is not active until the levels of M-cyclin increase. As the concentrations of M-cyclin increase in the cytoplasm, we have the potential to activate MCDK. However, to be fully activated, MCDK must be phosphorylated at an activating site by CDK activating kinase. CDK activating kinase phosphorylates MCDK, and if it's bound to M-cyclin, it can be active if there is not an inhibitory phosphate present. In order to get rid of that inhibitory phosphate, some amount of MCDK is able to phosphorylate CDC25, that is a phosphatase. Phosphorylation of CDC25 by MCDK results in its activation such that the inhibitory phosphate can be removed. In addition, active MCDK is also able to phosphorylate we. Phosphorylation of we inactivates the kinase, thereby preventing it from phosphorylating other MCDKs. So there are effectively two positive feedback loops. We get some active MCDK, and that MCDK is able to turn on a phosphatase that removes an inactivating phosphate, and it's also able to turn off the kinase that puts on the inactivating phosphate. It is these positive feedback loops that are responsible for the rapid increase in MCDK activity that we observe. Now, there are a number of ways that, or a number of places along the cell division cycle where the activation and the inhibition of cyclin-dependent kinases comes into play. On the top part of this slide, you see that CDC25 and anaphase promoting complex are responsible for activating or deactivating MCDK. I want to transition a little bit to SCDK and talk about inhibition of entry into S phase, because yet another mechanism of regulation is involved. So remember that S-CDK is going to be active during S phase of the cell division cycle, and it's during S phase that the DNA is replicated. Imagine, however, that you have damaged DNA. In this image, the damage was caused by X-rays, so we have double-strand breaks in our DNA. We don't want to replicate broken DNA, so we want to delay transition from G1 into S. The mechanism by which that occurs depends upon a protein known as P53 on the left-hand side. P53 is a protein that is continuously made inside of the cell throughout the cell division cycle. And if we need it, what we're going to do is we are going to prevent its degradation so that it can build up. In order to prevent P53's degradation, it needs to be phosphorylated. So by some unknown mechanisms or some very complex mechanisms, when we have damaged DNA, P53 becomes phosphorylated, and phosphorylated P53 is no longer degraded. It's stable. It is active, and P53's activity is as a transcription factor. Active P53 can bind to DNA, and it is responsible for transcribing a specific gene by binding to the promoter region of a specific gene known as P21. Transcription of P21 results in P21 RNA that is translated to produce P21 protein. P21 protein happens to be an inhibitor of S-CDK S-cyclin complex. So damaged DNA results in the production of an inhibitor of the S-CDK that would be responsible for initiating replication of that damaged DNA. So this is sort of an up close and personal picture of the same thing. We have active S-CDK, but when the DNA is damaged, P53 is responsible for transcribing P21 gene so that P21 protein is produced. And once P21 protein is produced, it acts as an inhibitor of the S-CDK S-cyclin complex to prevent progression of the cell division cycle until the damaged DNA can be repaired. So to summarize, in order to fully activate any cyclin-dependent kinase throughout the cell division cycle, it must be bound to a cyclin protein, a specific cyclin protein. It must be phosphorylated on an activating site, and for mCDK, this is accomplished by CDK activating kinase. It must also be non-phosphorylated or dephosphorylated in an inactivating site. For mCDK, this is accomplished by CDC25 phosphatase. And finally, we cannot have a cyclin dependent kinase inhibitor bound to the complex. And one example of a cyclin dependent kinase inhibitor is P21. We've spoken about progression through the cell division cycle as cyclin/cyclin-dependent kinase complexes have their activity regulated. We focused in upon the activation and the inactivation of mCDK, and we briefly spoke about the inactivation of sCDK in the presence of DNA damage. I want to introduce this one final player by focusing on G1-sCDK. G1-sCDK is responsible for transition over that restriction or start site, that particular checkpoint, in order for the cell division cycle to begin. In order for this to occur, there must be sufficient room to grow, so we cannot have that density-dependent or contact-dependent inhibition. We must have sufficient nutrients, and for eukaryotic cells, we must also have growth factors present. In addition, cells must also be adhered to the floor. They must be bound to the substrate in order to undergo cell division. All of those signals come in to tell a cell that it's appropriate to divide. What regulates that? What regulates this are the growth factors that we spoke about just a couple of weeks ago. Specifically, we're talking about growth factors binding to a growth factor receptor, and we learned that most of those growth factors are going to signal through our MAP kinase pathway. MAP kinase is going to be activated by RAS. Then that will activate MAP kinase, which in turn will activate MAP kinase. MAP kinase will go on and phosphorylate things, one of which is G1S CDK. Phosphorylation of G1S CDK activates it so that it can go on and phosphorylate other downstream targets. One of those downstream targets happens to be the protein called RB. In its non-phosphorylated form, RB is active. Its activity is as a repressor protein, and if you watch the optional video for last class period, you know that the transcription factor to which RB binds and inactivates is known as E2F. RB represses E2F's activity when it is not phosphorylated. However, when active G1S CDK at least partially phosphorylates RB, it must let go of E2F. E2F's transcription targets are genes responsible for promoting cell division. So in the presence of phosphorylated RB, E2F transcribes those growth-promoting genes or division-promoting genes such that the cell is capable of dividing. If G1S CDK had not been able to phosphorylate RB would have remained active as a repressor to prevent cell division.